In this final screencast for Chapter 2, we'll cover two families of organic molecules, the proteins and the nucleic acids. The third family of organic molecules I'd like to discuss are the proteins. Unlike carbohydrates and lipids, proteins contain a lot of the element nitrogen. The nitrogen is present in the form of amino groups. Proteins are polymers made from long chains of amino acids. There are more different kinds of proteins than any other type of molecule in your body. As you might guess from the diversity of kinds of proteins, they play a wide variety of different roles in your body. Among proteins' functions are structural, movement, and enzymes. As we saw with starch and cellulose, shape is very important in determining the physical and chemical properties of organic molecules. This is certainly true for proteins. Proteins are polymers of amino acids. So, what is an amino acid? As the name indicates, at one end of the molecule is a nitrogen-containing amino group. Bonded to the same carbon, but opposite the amino group, is an acidic carboxyl group. All amino acids share the same basic structure. A central carbon atom, which has an amino group attached to one side, and the acidic carboxyl group on the other. How amino acids differ is what else is attached to that central carbon atom. It can be as simple as a single hydrogen atom, or much more complex. It's been estimated that there are well over 1 million different proteins in the human body. All of these proteins, and all that we know about in other living things as well, are made up from the same 20 amino acids. Of these 20 amino acids, 9 are identified as essential amino acids for humans. By essential, what we mean is that they're required and cannot be synthesized by your body. Therefore, they must be present in your diet. These are a few examples of amino acids. Notice the amino group and the carboxyl group that are attached to the central carbon atom at the top of each. They differ in what is attached below that carbon. Small sequences of amino acids are called polypeptides. Poly means many. Peptide refers to the chemical bond that links each amino acid to its neighbor. More specifically, peptide bonds are the covalent bond joining the carboxyl group of the first amino acid to the amino group of the following amino acid. Guess what? You are already familiar with the chemical reactions used in the synthesis and digestion of polypeptides and proteins. Our old friend condensation synthesis is used to create a peptide bond between two amino acids. The OH from a carboxyl group of one amino acid and the hydrogen from the amino group of the second amino acid are split off, forming a molecule of water. A new covalent bond then forms between the carbon and the nitrogen atoms. It's this bond that we call the peptide bond. Plant or animal protein in your diet is broken down using hydrolysis. A molecule of water is inserted, breaking the peptide bond. Denaturation is a change in the shape of a molecule. Changing the shape of a molecule can have profound effects on its chemical and physical properties. Let's look at two examples. Protein is abundant in egg white. When raw, egg white is liquid, nearly colorless, and transparent. When cooked, the proteins denature, changing shape. The shape causes the egg white to become a solid, it changes to a white color, and it becomes opaque. In this example, the denaturation is permanent. In sickle cell anemia, the protein hemoglobin, which is found in the red blood cells, changes shape. In the presence of oxygen gas, the protein is a donut shape. When oxygen is removed, the protein becomes sickle-shaped. This change in shape of the protein also changes the shape of the red blood cells. They go from looking like jelly donuts to jagged, somewhat sickle-shaped cells. In this case, the denaturation is easily reversed by adding oxygen. Our fourth family of organic molecules are the nucleic acids. They're polymers made from nucleotides. DNA and RNA are examples of nucleic acids. Like amino acids, nucleotides contain the element nitrogen. But unlike amino acids, the nitrogen is present in the form of 5 or 6-sided rings called nitrogenous bases. These nitrogen-containing bases are attached to a 5-carbon sugar. Attached to the other end of the sugar is one or more phosphate groups. This diagram represents a simplified picture of a nucleotide. In blue is the nitrogenous base. The green pentagon represents the 5-carbon sugar, and the yellow circle with a P in it is the phosphate group. Only 4 different nucleotides are used to make all DNA. These nucleotides differ in which nitrogenous base is present. This figure represents a close look at a short segment of DNA. In the box is a single nucleotide with thymine, abbreviated T, as its nitrogenous base. The other bases are adenine, symbolized by an A, cytosine, C, guanine, G. Notice that A and T will form hydrogen bonds with each other, and so will C and G. A nucleotide which you may get tired of hearing about is ATP, adenosine triphosphate. Unlike the nucleotides that make up DNA, ATP has 3 phosphate groups. ATP is used to supply energy for many of the reactions which occur in the bodies of living things. The usable energy is stored in the bonds between the phosphate groups. Typically, just the bond between the second and third phosphate group is broken, releasing stored energy. The ATP is thereby converted to ADP, and a phosphate group is released. The reverse reaction is also possible. ADP can have the third phosphate group added, and energy is stored. This completes our second screencast.